So before I give the word to our facilitators, I think I will just give a few words of housekeeping and just a few instructions. So to all the participants that just joined, first of all, welcome, good morning, good afternoon, good evening, wherever you are in the world. You will see that you are all muted and that your cameras are off. You will be allowed to speak when it's time for the Q&A, so as per the program. But in the meantime, if throughout the presentations you want to ask a few questions, you will have access to the Q&A. It should be at the top of your screen. It's just a bubble that says Q&A, so feel free to write your questions there, as well as raising your hand. You have that possibility as well. If you would prefer to say your question out loud, that's also a possibility. Fortunately, you cannot unmute yourself because we have too many participants, so it would be a bit chaotic, but we hope that it will work with the hand raising and the chat. It's supposed to be very interactive, so feel free to ask questions whenever you want. And to answer the first question that I see in the chat, yes, all recordings will be made available to all the participants. So with this, I think I will give the word to Vanya to start the event and introduce everyone else. Thank you.
Thank you so much, Alessandra, and thanks, Marie-Noëlle, for leading the organization of such an important event. And thanks to all the facilitators. You all will see that it is a great team over the next few days that will be sharing some information and some content on very key topics for genetic resources conservation. We are very happy to have so many people interested in such a training. It shows how important it is and how we need to interact more and more among ourselves and learn together. We have in total over 400 people interested in participating who submitted their registration. We see that people are coming and coming all the time still. Today is just the first day. We will get used to the logistic of this tool. And among the 400 people, we have people from over 100 different countries. So it is a very diverse group. We are looking forward to interact with you all. As Alessandra said, it's going to be highly dynamic. We want people to talk, to make questions, to share your knowledge as well, to share your experiences, so we can continue building together this training. And the whole objective of all is that we can build a stronger global system for genetic resources conservation, that we are all part of it. Thanks everybody for being so interested in this event, and thanks for this huge team to agree on facilitating such a nice initiative. I would like to start by introducing our colleagues. So I'll call your names if you are here. I cannot see anymore only our colleagues. I just see a lot of people. So you please unmute yourself if you can open your camera and introduce yourself. Tell us your name, where you are based, and what you are going to be talking about. This is going to be good. Okay. So I'll leave you for last, Marie-Noëlle, because you are the most important. So I will start with Monica. Not because you're less important, huh? Just Marie-Noëlle is the outlier.
Thank you, Vania. Hello, everyone. My name is Monica Carvajal. I work at the Alliance of Bioversity International and CIAT based in Cali, Colombia. I'm part of the Genetic Resources Gene Bank. And yeah, I'm happy to be here. I will give you I think the first presentation. Yeah, thank you, Vania and Marie-Noëlle, for the invitation.
Thank you so much, Monica, for accepting being here with us today. We're very happy to have you. The second person would be Carolina Sansaloni. Carolina is the wheat gene bank head at CIMMYT. She's not here right now, but she's going to be here with us later. Then we have also Julian Soto from SIP. I'm not sure if Julian has connected. If
I don't
not,
see him. No.
If not, I'll move with our colleagues from CIB, Ronald. I think Ronald was connected.
Yes. Good morning. Hi, my name is Ron. I work at the International Potato Center in Peru, and I work in the potato collection. I will be talking about the second.
It's breaking, Ronald. We can't hear you well.
I'm sorry.
Yeah, your voice is breaking.
It's breaking.
Yes.
Mm-hmm.
So thank you, Ronald. I'll move to, let's see if Veronica's, Veronica, if we can hear you.
Yes, now it's better for me.
Now it's better, Ronald. Yes, please.
Mm-hmm. Okay. Hi, good morning. My name is Ronald Robles. I work at the International Potato Center in Lima, Peru, and specifically working with a sweet potato collection in the molecular studies. In the second day, I will be talking with you about the introduction of the DNA extraction. Thank you.
Thank you, Ronald. Veronica?
Well, good evening and good day for everybody. I am Veronica Valencia. I am a research assistant at Genebanks of CIB. I work as well as Ronald in everything that is about molecular characterization and bioinformatics analysis. So I will be speaking the second day too about DNA extractions applied to Genebanks. Thank you.
Thank you, Veronica. Rainer?
Good morning, good afternoon. My name is Rainer Vollmer. I'm on charge of the cryo program at the International Potato Center, and I will give you a talk about cryopreservation of clonal crops on September 9. Thank you.
Thank you, Rainer. So just so everybody knows, these first people who introduced themselves, it's now 6 a.m. in the morning for them. And thank you very much for your dedication and for being here with us today. Then I'll go to Julie.
So hello everybody, I'm Julie Sardos, I'm a genetic resource scientist. I work for the Alliance of Bioversity and CIAT. I am based in Montpellier in France. And so on Wednesday this week, I will present you on advanced data and more specifically on high-throughput genotyping data and the variant call format, otherwise called VCF. And next week on Thursday, I will also have a presentation on the ploidy with a specific example of banana, because I work with the banana collection. Thank you.
Thank you, Julie. Ranjana? Ranjana? She was here. We cannot hear you.
She's in the meeting. Maybe she's having connection trouble.
Yeah, I remember she was. Then I think Zach is also not connected. Zakaria is the gene bank head at Icarda in Morocco. Then we have Vetri from ICRISAT.
Hello, good morning, good afternoon to all of you. Myself, Vetri Vendan, genetic resources scientist working with ICRISAT, India. With me, Dr. Kuldeep Singh will be joining on Friday, and then Dr. Damaris will be joining. And then we will be talking on application of genomics in determining population size of germplasm, and then use of DNA markers for disease, and also the developing germplasm subsets like core and mini-core collection. This aspect we will be discussing on Friday. So nice meeting you all. Thank you.
Thank you, Vetri. Is Bart connected?
Should be.
I am. Yes.
Hi,
Hello.
Bart.
Hi. Hello, everybody. I am Bart Panis. I'm Gene Bank Manager of the Banana Gene Bank in Leuven in Belgium. So I am staff member of the Alliance of Bioversity International and CIAT. Next week, Monday, I will organize together with Rainer from CIP, who you heard before, a session on cryopreservation of plant germplasm. I will give an introduction, and then on Tuesday we will also do something together with an invited speaker on cryopreservation of recalcitrant seed species, and we will also discuss their requirements to set up a cryobank. Thanks.
Great. Thank you so much, Bart. Claudio.
Good morning, Vanya, and greetings to all the participants and colleagues. My name is Claudio Chiarolla. I work as legal officer with the Alliance of Biodiversity and CIAT, in particular for the Gene Bank Accelerator. We will be discussing policy issues, in particular on the session that will take place next Friday, so the last day of the training. I will be presenting together with Mathieu Rouard as well as Ruarit Hamilton about various policies and legal issues around access and benefit sharing, digital sequence informations, all data management related issues, why this has become an important aspect of the international negotiation that we are participating in and are affected by as CGIAR and as public research organization. So looking very much forward to discussing all these aspects with you, and we'll provide you, of course, of an update also about the Convention on Biological Diversity mechanism on sharing benefits from digital sequence information and related advancement in the negotiation under the plant treaty. Thank you so much.
Thank you so much, Claudio. Very important topics. We also have Cesar Petroli, who will be talking about bioinformatics. I don't think he's connected. It's 5 a.m. for him right now. And Mahinoel, if I didn't miss anybody, I give the word to you. You can also introduce yourself and yeah. And
okay, thank you, Vanya. I think I will, before I introduce myself, can I give the floor to Rure and then to Mathieu Roy, please?
Oh, they are both here. All right, I'm so
here.
sorry.
No
Since
problem.
Claudio talked about them, I thought they were not here. I'm so sorry, Rory and Mathieu.
No problem. Okay, so as you both said, yes, I'm Rory Sackville-Hamilton. I used to be head of the Erie Genetic Resources Centre until I retired from there in 2018. And since then, I've been helping Claudio and Michael and others in the policy group on these policy issues in relation to genetic resources in general, and specifically now on DSI issues. So I'll be there on Friday.
Thank you so much, Rory, for joining this webinar. And Mathieu?
Yes. Hello, everyone. So my name is Mathieu Rouard. I work with the International Banana Collection at the Alliance of Bioversity International and CIAT. I am based in France, and I will be talking next week on Wednesday during the Bioinformatics Day about data management for genotyping data, and also on the last day, the Friday, for policy on DSI, as Claudio mentioned. Nice to meet you all.
Thank you so much, Mathieu.
Vanya, now I can take the floor. Thank you, Vanya, for the introduction. So much. So good morning to all the participants. Good morning to the colleague. Good morning to friends. Good afternoon, wherever you are. I'm so happy to be here with all of you today, because this is something that we have been thinking of preparing, and to ensure that we will talk together on this important topic. So my name is Marie-Noël Ndionjob. I'm heading the Gene Bank at Africa Rice Center, and I'm also the interim leader for the area of work 5 on capacity strengthening of partners for enhancing ex situ and in situ conservation of plant genetic resources for food and agriculture. So I'm very happy to be here. During these 12 days, we will not just have a training, it will be more than the technical exercise that we go through. It is, to me, a strategic investment in knowledge, in innovation, and in collaboration. We should really, by advancing molecular characterization, we are not only equipping ourselves with powerful tools to understand the conservation and use of crop genetic diversity, but more effectively, we are also strengthening the very fundamental foundation of global food and nutrition security. So I will just now start with the introduction of the objective of this meeting, of this training, and then followed by the detail of the program that we will go through during the 12-day coming days. So I will share my screen now. Please tell me when you see my screen.
Yeah, we can see it.
So let me put it in the presentation mode. Okay. So thanks again and welcome to this training program. So the overall objective is really to deliver a conceptual foundation in molecular characterization, cryopreservation, bioinformatics, data sharing for clonal, legume, and cereal crops, to address also the application of digital sequence information in Genebanks and its relation to the policy on multilateral system. We would like also to prepare the participant for hands-on courses. We want the courses to be held in India and to fostering a community of practice on digital sequence information. So in the more specific way, this course will really strengthen, equip NAS scientists, our partner, with practical knowledge on DNA extraction, PCR reaction, genotyping, sequencing for clonal, legume, and the cereal crops. We will demonstrate the approach for conserving clonal crop and recalcitrant seed, ensuring the long-term genetic resources security. We want to train the participant to navigate on open access databases, analyze SNP, and apply digital sequence information in Genebank management. We would like to showcase how molecular data can guide diversity analysis, regeneration, gap analysis, and subset development for collections. We would like also to identify weaknesses. So like the 38% of the participants who like gene extraction, knowledge in gene extraction, or the participants who like the capacity on bioinformatics through the different sessions we will go through. We would like also to identify and mentor most engaged participants for the selection into the practical training courses that we would like to do by the end of this year. So the program will be in 12 days, almost 2 weeks. In the first week of the program, we will have the foundation of molecular characterization. And in the second week, we will have mostly, we talk about digital tools, bioinformatics, and then the policy dimension. So in more detail, we will have the first session today on molecular biology basics, the structure of nucleic acid, DNA, RNA, overview of molecular tools used for analyzing the genetic diversity. Then we have the presentation on genotyping, sequencing platform, the overview of available technological platform, the introduction of the concept of digital sequence information, and we also have discussion on the application of this technology. Then we will follow by the fundamental issue on DNA extraction, how to prepare the sample preparation in the gene bank, the protocols, the optimization, how do we also look at the quality of the DNA, because we need to how to assess what are the problem, when you face the problem, how do you solve them, and some guidelines. Then we will follow we will start the presentation on genotyping, how the data are generated. Of course, before that, we have the PCR reactions. So we talk also on the digital sequence information in the gene bank through diversity analysis, data handling, collection management. So we talk about that with a specific example on the regional perspective. Then we will talk about the gene plasma management, how to conserve the original genetic, how to determine the genetic structure of your collection, and how also to determine the optimal population size in terms of regeneration. Then we will start with the cryopreservation. Cryopreservation, we focus on clonal crop, and we talk about cryopreservation for recalcitrant seed as well. Then, most important things also, basic on bioinformatics. The bioinformatics is very important, the concept, the data management, how do you have access to open database, etc. And we are lucky that we have also a session on ploidy analysis. We have some introduction, general information, and then we focus on some specific crops. So the second week will be mostly on the access and benefit sharing. We will talk and the relation with GSI and how GSI became a key international topic, and the policy impact, the implication of access and benefit sharing under the CBD and also under the plant treaty. Then after all this presentation, we will also have we would like to have the participant about the causes analysis, we discuss about the key lesson, and then how we can, from there, we will start building this community on GSI. So what do we expect from you, participant? Please, we would like you really to be present and be engaged, to attend all the sessions every time and stay active throughout the courses, because we were able to mobilize the best scientists in this domain in the world. So we would like you really to be exposed, to ask the question, to raise the question, so that we can really start discussing on this important topic. You know that the genetic resources are related, also have the policy side. So we like to clarify all the question that you have in mind related to the policy aspect of the genetic GSI and genetic resources. So we would like you to contribute to the question, to the reflection and response to the discussion. And we would like also to bring in challenges and practice from the Gene Bank to enrich the dialogue. We are expecting you also to exchange experience and build network with peers from other countries, and also to connect the lesson on molecular tools, cryopreservation, and GSI to your institutional context. And we would like also to exchange experience and build network with peers from other countries, and also to actively demonstrate readiness for selection to the advanced practical courses will be held in India. So from this series of webinars, we would like to really to be the first step to introduce the practical courses and to build the committee of practice, whereby you'll be able now to talk together, to exchange knowledge to solve the issue by discussing among the network of the network that will be built. Okay, with that, I really want to thank you for your attention. So now I would like to give the floor to Alexandra for the follow-up. Alexandra, you have the floor.
Yes, thank you, Marie-Noëlle, and thank you for the presentation. I think it gave a great overview of our goals for this webinar. So I think what we'll do now, because it would be great to get to know also our participants besides our speakers, I think we will do a Mentimeter, if everyone's okay with it. So I will share the link to that in the chat, and in the meantime, I will share the screen so people can connect also with their phone. So let me share my screen here. So you should be able to see the Mentimeter now. So you can either connect with the link that I shared in the chat or with the QR code that you can see here. And once I have a few more connections, I will proceed with showing the questions. Great, we have around 37. It's going by super fast, so I'll just go to the first question. Anyway, you will still have the link in the chat if you want to keep joining. So here, we'll start with an easy one. This is purely for our own knowledge, just for statistical purposes. We want to understand the split in the gender of the participants. Okay, not really a 50-50 split. We have more female attendants compared to male attendants. I'll wait a few more minutes. Okay, thank you so much. Let's go on to the next question. So here I would just like you to pin more or less where you're joining from today, just so we can have an idea. We know the time is not super friendly for Latin America, so thank you to all the colleagues in that area of the world that are joining us really early in the morning. We appreciate the commitment. This is great. We're really covering almost the whole world. Yeah, I think we have a good spread. Here we can see a lot in Asia and Africa. More responses are coming in. Feel free to continue responding. I will move on to the next question. So here we want to understand which discipline you're specialized in. So we know that most of you will be specializing in gene banking or breeding, but surely we're going to have some other categories. So it would be great to know if you could write in the chat what you do if you're in the other category. Great. So we have mostly gene bankers with us. Okay. I'll move on to the next question now, just to get to know you a little bit more as well. So what is your title? Here you can type freely. Oh, not sure what happened there. Sorry. Here. Okay, so a good mix of research, scientists, biologists, technicians, conservation officers, breeders, as we predicted, some students, which is nice, researchers. Okay. A lot of biologists I'm seeing as well. That is interesting, as well as seed scientists. Agricultural engineer. Thank you. We'll move on to the next question. Have you attended previous CGIAR webinars before? Okay. A lot of new faces then, Marie-Noël, on this one. This is good. Then we should probably organize more so we can have more participations from people that don't really know us that well. Great, good to know. Thank you. Now a more general question. What motivated you to participate in the webinar? We'll have a question later on also about your preferred topics. So here you can be a bit more general if you want to. The aside. Genotyping, gene bank management. Okay, career change, career development. So this is also for, let's say, professional growth. This is great. We'll hope this will bring the knowledge you were looking for. I'm sure it will. Cryopreservation. So some people are also part of our communities of practice already. So they were invited automatically to be part of this. This is great. Bioinformatics, molecular analysis. Great. I'm really happy to see that the topic was interesting to you. I mean, we saw it from the amount of signups for this webinar. Bioinformatics, yeah, that's a topic for later in the webinar. Personal development as well, great. So what is the biggest challenge you face related to the topic of the webinar? This can be one word, just the reason why, the challenge that motivated you to join this webinar. It could be a particular topic you're not so familiar with. It could be that you don't really know the CGIAR gene banks and you want to get to know them better. Or a challenge you face in your everyday work that is something you're missing. Okay, so bioinformatics is a big one. Mathieu, this is for you. Computational tools, data analysis. Okay, most people are interested in bioinformatics and it's a challenge for them. So it's great that we have some of the best scientists in this webinar to talk you through it. Cryopreservation is also a word, so it's good to know. There'll be a big focus on it next week. Great, thank you. So finally, well, we know bioinformatics is already at the top of this list, but what topics are you most excited about for these next 2 weeks? Just to understand a little bit where the needs are for the group. Okay, data. Okay, so bioinformatics, as we know, is at the top. We have molecular characterization data. We know cryomploidy can be a bit specific, so it makes sense that they are not so key in this list, but that doesn't mean that they're any less important. Okay, this is great. Thank you so much for your answers and the time you put into this. It's great to know your attitudes towards the webinar. So I will now stop sharing my screen. And I will leave the word then to our first speaker of today so that she can deliver her presentation since it's very early in the morning. So Monica, if you're still online, I will give you the word. You can share your screen, deliver your presentation.
Hi, yes, I'm here still.
The floor is yours.
Thank you. Can you please confirm if you can see the screen?
Yes, and just a quick note for the participants, if you have any questions for Monica, please note they will be addressed at the end. There is one Q&A for both presentations today, and use the Q&A chat. Thank you.
Okay. Oh, hello everyone again. Good morning, good afternoon, good evening. Yes, so it's a pleasure to be in this webinar series and to be the first one to give the presentation. I will talk about exploring the molecular applications for regional priorities. My name again, Monica Carvajal. So I hold a bachelor degree. I have also a PhD in virology with focus on molecular and cellular biology. I joined the Alliance of Bioversity International and CIAT in 2012. And I'm currently the team leader of the Digital Gene Bank, which is one of the teams of the Genetic Resource Program. So we implement genomics to reveal the genetic structure of the collections that we conserve, to enhance the conservation and to promote the use of the crop diversity. As I mentioned before, I'm based in Cali, Colombia, and this is the team I work with. So for the presentation, I will divide this in two parts. The first one is to review the basics of the structure of nucleic acids. So I think probably all of you are familiar with this, but just to ensure that we are all in the same level, we will also take a look of some molecular tools, not in depth because there will be different presentations to go in depth on some of the molecular tools. And we will also explore what is the role for genetic diversity analysis. And the second part of the presentation, I will dedicate to show a case study of how in Latin America we are leveraging molecular applications for tackling regional priorities and also to enhance the collaboration. So let's start with the first part. So the nucleic acids are linear polymers of nucleotides. These are large biomolecules, and they are the nucleotides are the building blocks for the DNA and RNA. So this monomer that you can see here in the diagram is composed by a 5-carbon sugar represented here in blue, a phosphate group represented here in green, and a nitrogenous base. The two main classes of nucleic acids are the deoxyribonucleic acid, if the sugar is a deoxyribose, and the polymer is the DNA, or a ribonucleic acid if the sugar is a ribose, and the polymer in this case is the RNA. So as you can see in the diagram, so the only difference between these two sugars is the presence or absence of a hydroxyl group in the carbon 2 of the sugar. So the bases that are part of these nucleotides are found in RNA, and the DNA are the ones that I just showing here in the slide. So they are adenine and guanine. These are two double aromatic rings, also known as purines. They are also cytosine, thymine, and uracil. These are single aromatic ring molecules, and they are known as pyrimidines. And as you can see, the thymine only occurs in DNA molecules, while uracil only occurs in RNA molecules. So the strings of nucleotides are bonded to form these spiraling backbones that you can see in the diagram. They are assembled into chains of bases to form these single-stranded or double-stranded molecules. So the sugars and the phosphates in the nucleic acids are connected to each other through phosphodiester bonds around the outside of the helix, represented here in green. And the nitrogenous bases point towards the center of the molecule. So the DNA these two chains are held together by hydrogen bonds between a specific pair bases. Each base pair consists of one purine, either adenine or guanine, and one pyrimidine, the cytosine or thymine. So this pattern is known as the complementary base pairing. So you can see in red the adenine pairs uniquely with thymine by forming two hydrogen bonds, as you can see here represented. While guanine in blue pairs with cytosine in green by forming these three hydrogen bonds. So this structure is, I mean, I know it's basic, but it's good to refresh because this will, for example, for the PCR reactions, it will be relevant in terms of designing primers and the profiles for running the PCR. The direction of the polynucleotide can be defined at the linkages of the phosphodiester bonds between the adjacent nucleotides. So the prime and the blue numbers around the sugars designates the position of the carbon in the 5-carbon sugar, the oxyribose of the DNA. In the sugar phosphate backbone of the DNA, the phosphate group that connects the 3' carbon of the sugar with the 5' carbon of the next sugar is linking successive sugars together. You can see with the blue arrow, the 5' and with the black arrow, the 3' of the sugar that are connecting. The ends of the polynucleotides, specifically the 5' phosphate group, at one end is called the 5' end, and the 3' free hydroxyl group at the other side, here highlighted with the black circle, is called the 3' end. So these two strands run into opposite directions, forming an antiparallel molecule, which is characteristic of the DNA. So now we refresh the basic structure of the DNA and mention also some of the RNA. And we know that the DNA is the manual, the instruction manual, because it carries the information in cells, it encodes and stores information in every living cell of every life form on Earth. So we know that the genes in the DNA are also the instructions for making proteins, which in turn determine many traits, such as plant height, seed color, disease resistance, among others. So the RNA is this messenger of information. However, from the total DNA, it is estimated that around less than 3% of the DNA encodes RNA for making proteins, while around 80% of non-coding DNA produces different types of RNA molecules, and these are known as non-coding RNA genes. So these DNA elements, they code for RNA molecules, but they don't become protein. However, they play a role in regulating, they play a major role in regulating when and how much a protein is produced. The other percentage of DNA in cells that you can see in orange correspond to other parts, other regulatory elements or other structure elements like introns, telomers, satellite DNA. These satellite DNAs are high repetitive sequences of DNA which are scattered through the genome among other elements in the DNA. So we also know that the DNA can harbor variations, which are also known as genetic variants or DNA variants, which are the differences in DNA sequences between individuals. So these variations can arise from mutations during the DNA replication, exposure to environmental factors, but they can also be inherited from parents by recombination. So they contribute to the diversity of traits and the characteristics within population and can influence individuals' response to different stressors such as diseases or stress in climate and some others. So the type of variations are summary here. So there are some point mutations when a single base of is altered. There are also insertions or deletions known as indels when the bases are added or lost. And there are also structural variations when there is a translocation, inversion, and deletions or duplication, but in this case is when large DNA segments are involved. So these variants can alter the protein structure and therefore also their function. They can also determine how much a protein is made and also when and where a protein is produced. So there are different impacts on these DNA variations. Identifying the DNA variations is relevant because they can help us explain why individuals, populations, or species they differ in their traits, as I already mentioned. So these changes can affect traits such as yield, nutrition, quality, disease resistance, among others. But they play also an important role in evolution and adaptation to climate change. So identifying these variants have also applications in conservation, as you will see through these different series of presentation in the webinar. They can help distinguish unique accessions and detect duplicates in the collections, and this can be very helpful for managing and optimizing conservation efforts. But they can also ensure that the rare alleles can be safeguarded for future generations in the gene banks. They are also relevant in breeding because they can be used as molecular markers and allow breeders to track inheritance of desired traits. They can help select superior plants and also help to introduce useful genes from wild relatives into cultivated varieties. But they can also provide some insights, as I already mentioned, into evolution and domestication. So the molecular markers can help to detect genetic variation. We know that now. There have been different kind of molecular markers from protein. They are protein and DNA-based markers, and each class offers a series of opportunities and challenges. However, as you can see in this diagram, over the decades, various molecular markers have been used to detect and characterize genetic variation for the purpose and that some of the purpose that I described in the slide before. So you can see this diagram is taken from a publication from Nature Reviews, and it shows how protein-based molecular markers were most popular until the late 90s. In recent years, of course, because the advances in sequencing technologies, the DNA-based or sequence-based markers have been expanded, and they are now the most used ones in the last decades. So I won't go into detail into this diagram, but I just want to show, I think this diagram summarizes very well the different DNA markers, noting that the DNA, the PCR-based markers are the ones with a broader offer. I think during the seminar you will review some of them in detail, but for now I just want you to take the message that there is a big offer of molecular markers, specifically DNA markers based on PCR, and depending on the purpose, the technical capacity that is available, and the resources, one can select the one that fits most the purpose. So these different molecular markers that have been developed over time have been used for different studies, and they have been applied for genetic diversity and population structure studies. So I won't go into detail, but I just want to let you know that there is a lot of publications that are available that detail the use of these different applications for genetic diversity analysis. But I will just share with you briefly some brief examples of the diversity analysis that we are conducting in future seeds. which is the Alliance Gene Bank. I mentioned that I work there, and we conserve the world's largest collection of cassava, bean, and tropical forages. And we are implementing genomics. Specifically, we are also identifying DNA variants to help us reveal the genetic composition of our collections. So to achieve this, of course, we will have to follow a series of steps, and you will review, I think, all of them through different presentations by the colleagues. This includes tissue collection, DNA extraction, and the different ways that DNA variations can be detected. Some statistics or that needs to be performed, like estimation of genetic distances and population, and how to visualize the different groups that the materials that are conserved in gene banks can form to help us understand the composition of the collections, among others. But I just want to share that in our case with the cassava collection, you can see in this, we conserve cassava in vitro, and this pie chart shows the composition of our collection accordingly to the passport information. But after running genetic distances and population structure analysis, we can see in this multidimensional scaling how our accessions, our cassava accessions are related among themselves. So same color, blue and red represents cultivated accessions, while green are the wilds. As expected, they are farther apart because they are less related to the species, the cultivated species. But it helps us understand whether what we are conserving really reflects what we think they are. But we can do more than just understanding our collections, so we can compare between collections. There is another CG center that conserves cassava diversity, and this is IATA, which is based in Nigeria. They conserve most of the cassava, the African cassava diversity. So taking advantage that the two centers use the same platform to detect these DNA variants, we use genotyping by sequencing. We had the opportunity to compare our collections, and you can see that in green is a CIAT collection, in orange is IATA collection. We can understand that the two collections are complementary. There is very few overlap, so it is relevant for us to keep these two collections separately. And if we want to infer a bit more of the rationale of why these two collections are separated, is because when cassava was introduced to Africa, it faced different challenges than the ones that cassava evolved in South America. So meaning that breeders start to introduce introgress resistance to a disease using a wild relative which was not previously related for the South American germ class. So this is one of the reasons why it's so different. And it's good to understand, because it's good to know if how to use the efforts for conservation. So I believe, and I strongly believe, that the molecular applications matter for regional priorities and can be used to strengthen regional collaboration if, for example, joint diversity analysis are conducted together. So now I will switch to the second part of the presentation, and this is about how we are leveraging molecular applications to tackle regional priorities and to enhance collaboration. And I would like to present a community of practice that was established since 2021 in Latin America. This has been a work of, together with the Alliance, CIMMYT, and these are the people that has been working with also support from SIP. And it was catalyzed initially by Diversity International, DIFSIC, and it has been supported by the Gene Bank initiative as well. So why we want to empower gene banks in genomics? So Latin America and the Caribbean are primary centers of domestication for staple crops, which are very relevant for global food security. This includes crops such as cassava, common bean, maize, potato, among others. but they are very significant because they support as a staple the world's population. Moreover, in Latin America, there are around 200 gene banks. They conserve over 5,000 cultivated and wild crop species, and they conserve close to 500,000 accessions. So we also know that despite the advances in genomics, national gene banks in low and middle income countries, including those in Latin America, face persistent barriers to effectively apply and implement these molecular tools for enhancing conservation and promote the use of the collections. Some of the challenges which might be similar to other regions are, for example, the sample handling, limited capacity in data analysis, the high cost for purchasing reagents and equipment, procurement difficulties, among others. So that's why we believe that the strengthening the cooperation and on emerging technologies for characterization, conservation, can really enhance the effectiveness of national gene banks and ex situ collections. So similarly, as some of you, I seen that some of our participants of the community of practice joined this webinar, and we are very happy that they are also being able to learn from this webinar. But similarly, as it has been done, I think for the preparation of this webinar, we organized a survey to collect some information to help us identify the regional specific needs, the capacity gaps, and collaboration opportunities in Latin America. But regarding the use of genomics and digital tools to support gene banks collections. We also take advantage of the survey so that we can update our contact information. So we know that in the last years, there have been a generation exchange. So new people arriving to work with genetic resources. And also we wanted to learn what are the genetic resources that are conserved in the region, what is the size of collections, and if they have used already some genomics for gene banks, what was the main purpose. The survey consisted of 34 questions responded by 48 different people from 14 different countries affiliated to 33 institutions. Of course, I won't show every question. I just want to summarize that one of our main questions was if they want to participate in a community of practice, what will be the preferred language? In Latin America, most of the countries speak Spanish, but there are a few that speak other languages. So we needed to make this question. At the end, we are the community of practice is in Spanish. We, as you can see, so how many accessions does the collections have? Most of the responses were between 1,000 to 10,000 accessions, whether they have already genotyped some of the collections. So we have a broad range of responses, but most of the responses were very few, between 0 to 5%, or between 0 to 20%, let's say. And the purpose of genotyping was mostly genetic diversity analysis, germ plus characterization, and trait discovery. So another part of the questionnaire was to prioritize topics for developing capacity and workshops. So these in yellow are the four priorities that the community decided. So working on analysis, interpretation, curation, and storage of genomic data, followed by genotyping platforms, then the use of tools and applications for enhancing sample traceability, for generating this genomic information, and then also the policy framework regulating the use and access of genomic data linked to or derived from gene bank collections. So to address the top one priority, the analysis, interpretation, and curation of genomic data. So we did a follow-up questionnaire asking whether they have used a specific platform for genotyping, for identifying these DNA variants, called DARTSEC, that Carolina probably will give some insights later in her presentation. And we noticed that most of the participants have not used this platform. So we thought that this is an opportunity to learn on this specific platform to address specific challenges. And one of the main, so we then ask for research questions that are relevant after generating genomic data jointly. And 70% of the participants mentioned that one of the interests is to understand their collections in relation with other collections from other countries. We also have some responses like interest in knowing what are the collection gaps in their collections, but also whether there are duplicates in their collections. So now having knowledge on what are the needs, we developed through the years a series of virtual and in-person meetings where we address the different priorities. Then in 2022, we had the opportunity to use this platform that I just mentioned, DARTSEC, to generate genomic information from 5 crops that were selected by the community, including beans, potato, cassava, maize, and wheat. And countries provided some samples that they were genotyped, collectively having like this group of samples per countries that are part of the community. During 2023, countries reviewed their own data, and then they co-analyzed the data with other countries and performed some analysis. And then between 2023 and 2024, we have been meeting regularly to discuss the results and think on what, in addition to understand the composition of their collections and their collection in relation with other collections, what other things we can do together. And really interesting things came out. Carolina will give a presentation during this week about some details. I just want to summarize that with this exercise, with the community of practice, we have been able to promote new collaboration between institutions using molecular applications as a starting point. And here you can see some pictures of the meetings, of the people participating in the community, but also below some maps. And these are the 3 maps of the 3 different crops where institutions have been bringing together their samples for analyzing. So we have for being 7 countries and 9 institutions. Some were able to bring samples and genotype. Some others, they haven't, for multiple reasons that we are also discussing and trying to address. And maize, 5 countries and 7 institutions are contributing to the joint analysis, diversity analysis, and for potato, 3 countries and 4 institutions. So as I mentioned, Carolina will provide some insights on the case of beans. And this is a very nice picture of our members of the community discussing some of the results they obtained in the diversity analysis. Yeah. So I think this is all for now. So just to close, so we have seen the very basic composition of the nucleic acids, how they are the foundation for molecular analysis. Identifying variations in nucleic acids are the basics to bridge the basic science with applied breeding and conservation strategies. So the molecular tools can enable precise diversity assessment. And I'm very happy to share the Latin America community of practice, how we are leveraging these tools to resolve some of the challenges, propose regional impact through regional core collections, for example. So this is all from my side. Thank you very much for your attention, and I'm open if you have any questions.
Thank you, Monica. Marie-Noëlle, over to you.
Thank you very much, Monica, for this excellent presentation. We will now give the floor, like I told you before, and Alexandra also come say the same thing, is that we will please keep the question for the QA session at the end of the two presentations. So without further delay, we will now give the floor to our colleague Carolina. So please, Alexandra, if you can put her presentation on, because it's like 5 a.m. in Mexico, and Carolina is not feeling well today. And so we listen to her presentation, but she will be there for the question and answer session. Thank you, Alexandra.
Yeah, no problem. If I could ask everyone to raise their volume a little bit, because it is a recorded presentation and sometimes the audio can be a little bit low. So just sharing my screen. Let me know if you can see it. And I will.
Yes, please put it on the presentation mode, please. Yes, great.
Or afternoon, depending on the country you are now. I'm Carolina Sansaloni. I'm currently a scientist at CIMMYT. I joined CIMMYT in Mexico 13 years ago, working in a high-throughput genotyping platform, mainly for characterization of genetic resources, but also with breeding program. And 4 years ago, I joined the Genebank, working as a Genebank manager of the WIP collection here at CIMMYT in Mexico. Today, I will present an overview of the sequencing and genotyping platform and also digital sequence information, specifically for plant genetic resources. To start, I would like to share that this slide highlights the foundational role of genotyping and sequencing in modern plant science. First, they allow, why this matters really? First, they allow us to reveal genetic diversity, not just as cataloging variation, not just as a catalog of biodiversity, but also identifying unique alleles and reducing redundancy, for example, in Genebank's collection, which is super important for managing the collection. As I say, this is essential for maintaining the integrity and the value of conserved gem plants for today and for future generations. Also, they support both conservation and use. On the conservation side, genotyping could help us document diversity and monitor genetic integrity, for instance. And on the utilization side, it enables us to identify promising accession and link them to a specific trait that can be stress tolerance, could be heat, drought, nutrition, quality, and all these traits guiding the pre-breeding and research activities. And finally, these tools accelerate breeding innovation by enabling marker-assisted selection and genomic prediction. They shorten breeding cycle and increase precision, especially for traits like stress tolerance, yield, and the nutritional quality, as we mentioned before. So in short, genotyping and sequencing are not just technical steps. They're strategic enablers of biodiversity stewardship and crop improvement. And because this is a webinar for different level of scientists, students, I would like to start from the beginning, and I apologize for the more experienced colleagues, but it's important to understand the context of genotyping and sequencing. So in this slide, we illustrate the remarkable transformation of DNA sequencing technology over the past 4 decades, starting with Sanger sequencing that I hope many of you had the opportunity to run. I'm not even talking about gels. No, if we go a little bit further, the original sequencing was done on gels. No? So it was very hard work. Later, the Sanger sequencing in the 80s, And then we move through high throughput platforms like the Roche 454 and Illumina, which is currently one of the most used platforms. And into the current era of next generation and real-time sequencing tools such as PacBio or Oxford Nanopore. But alongside these technological leaps, we see an exponential increase in sequencing entries in the gene bank. Many gene banks are now characterizing their germ plants. And this is driven by global initiatives like the Southern Genome Project on the democratization of sequencing tools. And also on the right side, you can see the cost curve that tell another very powerful story. It's very impressive, the dramatic drop in sequencing costs that has outpaced even more law, making large-scale genomic study and personalized application more accessible than ever. This evolution not only reflects scientific progress, it reshapes how digital sequence information is generated, stored, and governed. As platform evolve, so do the responsibility around the data sharing, benefit distribution, and policy compliance, as we will see soon. If we use as an example the human genome to represent the evolution of the technology, we can think in 2001, when the Human Genome Project started, it took 11 years to complete the genome, and it cost $2.7 billion. Then technology evolved, and the time and the cost reduced significantly. Again, gaining the Moore law for far. Following by 2001 also using Celera technology, then the 454 from Roche, Avi Solid, then Illumina. And today, we can think in, well, not today, like few years ago, we started to see whole genome sequencing for $1,000. And today can be even less, $500, and in some cases even less, let's say 200, 300. Maybe not for a human genome, but for some species, it's a reality. So definitely the technology evolved super fast, reducing time and cost, and increasing, of course, the amount of data produced. If we do an analogy of the different sequencing generations, we think in the Sanger sequencing as an old-fashioned camera, photograph camera. I don't know how many of you had the opportunity to take pictures using this camera. I did. And it takes a lot of time to reveal the picture. It was very risky, and maximum of 24, 36 pictures, and that's it. Then we move to next generation sequencing with Illumina as an example, HiSeq sequencer. And if we compare 800 Sanger sequencing was the same than one HiSeq Illumina sequencer. And this we can compare with a digital camera in which we can download the picture in a computer and we can store thousands and thousands of pictures. But now we have a new technology, as an example, the NovaSeq X from Illumina. And if we compare it, 16 HiSeq, it's one NovaSeq X. So again, the amount of data produced is much more powerful. And now we can compare with the last iPhone that we can take thousands and thousands of pictures, and it's automatically stored in a cloud. can be shared. So it's much more easy and effective, the data generation. In all this evolution, the next generation sequencing, sorry, use micro and nanotechnology to reduce the size of sample component, lowering reagent costs, and enabling massively parallel sequencing reactions. That's why it's cheaper and it's faster and generate much more data because it's highly multiplexed and allowing simultaneous sequencing analysis of billions of samples at the same time. This timeline captures the convergence of plant breeding, market technology, and sequencing innovation from 1980 to today, 2025. We begin with the development of molecular markers like RFLP and SSRs. I don't know how many of you had experienced this type of molecular markers, were super useful at that time, which leads the foundation for mapping and QTL analysis in the 90s. As we move forward, we see the rise of NIPS, genotyping by sequencing technology, and now high accuracy long read platforms like PacBio or Nanopore. Parallel to these technical advances, breeding strategies have also evolved from GWAS analysis and genomic selection to CRISPR-Cas-based precision breeding. And also looking ahead, we can think in AI-driven decision making. So it's fascinating, the speed of the technology evolution. At the bottom, the cost throughput diagram tells us a compelling story. Sequencing has become dramatically cheaper and faster, enabling large-scale genotyping and accelerating crop improvement. So costs reduce and the throughput increase super fast. Well, to provide more information about the different genotyping platforms, as we mentioned before, we have the marker-based genotyping, like SSR or the famous macro satellites, which are very polymorphic with low cost, but limited coverage. Some applications are diversity studies and small scientific projects. Then we move to array-based platform, for example, a SNP or Dart array, that are highly reproducible and standardized protocol, but with fixed content and limited SNP. So depending on the objective, they are very good technology. Then we have the next generation based approach, like whole genome sequencing, which are very compressive and costly, GBS or DART-seq. These are cost effective for large implants. We also have RNA-seq, exome for functional regions, long read sequencing, the PacBio, high nanopore, structural variant, repetitive regions, and some applications are of course reference genome or even the pan-genome. But how we choose the right platform? We have today a lot of information, a lot of platforms available, a lot of companies providing different platforms. How we choose the right one? Well, it depends on different things. First, we need to understand the research question. We need to evaluate the cost versus resolution. We need to identify the genome size of the species, the complexity of the genome, if it's a polyploid species, if it's a diploid species, if it's a wide relative species, and also the available expertise within our team. So there are a range of information that we need before evaluating what is the right platform. So if we want a small project, maybe a macro satellite works perfectly well. But if we want to see the whole genome or discover diversity in a gene bank, maybe the next generation based approach are more appropriate. So we really need to evaluate all these different questions before selecting the right platform. I would like to focus now in the technology we selected at CIMMYT for the gene bank characterization. In this case, we use DART-seq technology. If you imagine, we have two large collections, two of the largest collection of maize and wheat, 28,000 accession of maize and 124,000 accession of wheat. And so it's a very large collection. And we choose DART-seq technology because it gives us an idea of the whole diversity of the gene bank without a fixed, for example, a fixed array or a fixed SNP chip. DART-seq technology is more appropriate for this type of diversity characterization. So in this graph, you can see we start from the gene bank, of course, taking a little sample, a few seeds per accession. We plant it in greenhouses, and approximately 1 week, 10 days, we harvest fresh leaf for DNA extraction. And then after the DNA extraction, we do the DART-seq technology. In this case, we do in-house in a lab called SAGA, the Genetic Analysis Service for Agriculture, that we use the DART-seq technology provided by Diversity Array Technology Company. But of course, not only DART-seq technology is good for characterization of gene bank. There is a big range of application of this technology in plant conservation and also in breeding. I presented before, and please, we will explain more in detail all of these topics during the webinar. So this is a very short summary, but don't worry, all these topics will be explained more in detail later on the day. So some of the application, as I mentioned before, is the genetic characterization and diversity analysis. In my case, for gene banks, it helps us to assessment of genetic diversity within among accession, identification of the population structure or phylogenetic relationships, also monitoring the genetic integrity in the gene bank collection. In marker discovery, high throughput SNP and silico DART markers are generated through this technology. And one very important thing is we don't need a reference genome. So for new species or rare or wild relatives, for example, that we, in many cases, we don't have reference genome, this is a very good technology to use. Detection of rare allele and polymorphisms, it's also super important. More in breeding, we can be used for QTL mapping for different traits, GWAS analysis for trait marker association, marker-assisted selection, of course, for a split breeding program, identification of markers linked to heterosis effect. Again, in conservation and germplasm management can be used in evaluation of genetic purity, redundancy of accession as well, optimization of sampling strategy for regeneration in the field, support core collection, and the development and curation decision. In genomic, more in innovation, in genomic selection and crop improvement, this technology can be used in genomic prediction model for accelerated breeding, integration, tracking of wild allele into elite lines in pre-breeding programs, enhancing genetic gain in crops with narrow diversity, and in functional genomic and epigenetics for methylation profiling and integration with transcriptomic and phenotypic data for system biology. So as you can see, it's a very diverse application using this type of technology. All the information generated using static or any other technology is currently stored in many databases and published in different scientific journals. So today it's relatively easy to access to all this information as well as generate new data. You can see a very high impact journal having a lot of information about whole genome sequencing or a big characterization project. So we discussed a lot about sequencing, genotyping platform, and the result of this are the famous DSI, the digital sequence information. And what is this? Well, refer to the data derived from the genetic material of an organism, specifically the sequence of DNA, RNA, or protein. In the context of plant genetic resources for food and agriculture, DSI includes genomic data that help researchers understand, conserve, or improve crop varieties. So starting from the seed, we extract DNA, we sequence using all the technologies that we discussed before. We store in a database, and this data can be used for breeding applications. So this information can be extracted from seed, tissue, whole plant, and stored in different databases for research and breeding programs. So what DSI or digital sequence information means for plant genetic resources? We have different areas. For example, in conservation, the DSI is transforming the way how Genebanks operate. Instead of relying solely on passport data or phenotypic traits, we now use DSI to deeply characterize the genetic makeup of accession. And this allows us to identify redundancy, detect genetic drift, and prioritize unique or underrepresented material for conservation. It's especially powerful for building core collection, for example, and guiding regeneration strategy with more precision. In genetic insight, DSI gives us a high-resolution lens into genetic diversity. We can pinpoint alleles associated with key traits like drought tolerance, disease resistance, or nutritional quality, for instance, and use that data to guide pre-breeding and parental selection. And this bridge the gap between conservation and utilization, turning stored seeds into strategic assets for crop improvement. But also as a global asset and equity, one of the greatest strengths of DSI is the digital nature. It can be shared instantaneously across borders, enabling collaboration between institutions, researchers, and breeders worldwide. But this also introduce complexity. When DSI is used commercially, for example, in developing patent varieties, how do we ensure benefit flow back into the countries or community that originally provide this genetic material? This is where CBD decisions come in, proposing a multilateral benefit sharing fund to address this gap. This topic is super important and will be discussed more in detail later in the webinar series by experts in the area. So don't worry, you will see more detail of this policy topic for DSI. This slide is very nice. It was published recently by our colleagues, and it represents the landscape of the genetic sequence database in the era of digital sequence information. Genetic resources are characterized using different sequencing technologies. The resulting sequence data can be shared, stored, and used in multiple ways, starting from the genetic resource that can be, in this case, talking about genetic resources for food and agriculture. We sequence using all the technologies I discussed before, and then can be stored in different, in multiple ways. Could be core public repository, a specialized database, secondary database linked to scientific publication, for example, private or commercial database. So it can be stored in totally different environments. So this data set can be accessed and downloaded by different users for different purposes, including research projects, breeding application, or policy-related studies. So this landscape will be further expanded and discussed in detail in the coming session, even from the author of this publication. As an example, just an example of how important are DSI in plant genetic resources. As a gene bank manager, we have thousands of accessions conserved in the gene bank, some of them very similar. Just by looking at a maize in this case, we can identify, okay, this maize is good for tortillas, famous tortillas in Mexico. This maize is good for pozole, another very famous dish here in Mexico. But we cannot see the traits hidden inside the seeds. For example, if it's heat or drought tolerance, if it's resistant to any diseases. So these traits are hidden inside the seeds. And only by using DSI, we can explore the potential of these accession conserving the gene bank. So in this picture, you can see same maize, but two different destinies. One, heat susceptible, as you can see, very poor plant, and of course, represent empty plates at the end. And on the other hand, we have heat tolerant maize with in the same climate, but it's tolerant to the same climate and can feed family and securing the future. So this is just a representation of how important are DSI in plant genetic resources for agriculture. So to finalize my talk, I want to say that DSI, digital sequence information, is key to unlocking the genetic potential of crops, enabling sustainable agriculture that can nourish future generations and ensure food security in a changing world and in a changing climate. So this is a funny picture to represent how was before this sequencing technology for germplasm conservation and utilization. Breeders use a lot of time. Imagine if you are a breeder and you want an accession from 124,000 accession gene bank. How to start? Which one we select? So it was super hard. Now the new way using DSI is more precise, is more effective. The search into the biodiversity inside the gene bank. So there is a big change before DSI and after DSI. With this, I really want to thank you for your attention. Again, all the topics we presented here will be developed more in detail for different experts in the coming days. So yeah, thank you for your attention. This is my email, so in case you have questions or you want to reach me for any particular topic, I'm more than happy to talk to you. Thank you.
Thank you. Thank you all for listening to the two presenters. So now we will start the question-answer session. There is no question, you just listen to two presentations, important one on the fundamental of the molecular biology. You talk about Monica talk about the nucleic acid. as a foundation for molecular analysis, or also how to identify the variation in nucleic acid within the population. She talked about the different variation that could be created by mutation, by deletion, insertion, translocations, and so the different molecular tools that enable precise diversity analysis. And then she provided example of how these tools help them to build the community of practice in Latin America, how they come to that level, and how now in Latin America they can collaborate and to address the regional issue, issue related to DSI or molecular tools. So then we follow, the presentation was followed by Carolina presentation mostly on the genotyping as a support, conservation and use. And she showed the evolution on the platform, how we start with the sequencing using Sanger method. It was in the early 80s, very difficult, but it was very good because it helped us really to move on, and how the platform have changed up to the next generation sequencing using nanopore. She also mentioned about the timeline of evolution of marker technology. We start with RFLP, rapid, SNP. Now we talk about genotyping by sequencing technologies. And now she ends her presentation by talking of application of that seq platform in plant conservation and also breeding. Then she also mentioned, give a specific intervention also on digital sequence information. Oh, Carolina is here. So I'm happy now to open the floor. Carolina and Monica are here so that they will respond to the question. So the floor is open. Please raise your hand and open, if it's possible, the camera to raise the question so that they will be able to respond to your question. Thank you. So there is one question in the chat, Carolina. As sequence technology is evolving rapidly, how easy would it be to club and analyze the old sequence sample and new sequence sample together?
Yes, thank you. And hello, everyone. I really apologize. I had to record the presentation because I'm coughing a lot, and I was afraid that I will cough during the whole presentation. So really sorry. It's a great question, and I think it's an issue that all scientists are facing now. If we can combine old sequence with new sequence, and I can tell you a few examples of from our side. We generate a lot of data with previous sequencing technology and develop molecular markers. And now we are using different technologies, same methodology. We are using DARSeq in this case for gene bank characterization. Sorry. But now we are able to combine this technology by having the raw data, and this is very important. We use the raw sequencing data, can be FASTQ file, and we compare previous analysis. We extract FASTQ file data from previous analysis and the sequencing data from new analysis, and we can recall the molecular markers again. So this has been working very nice. We have several examples doing this. So it can be done for some examples. Of course, we cannot compare super old technologies with the last technology, but just by reducing at raw data, it can be done.
Okay, thank you, Carolina. So thank you for this very elaborated response to the question. So I can see if there are some hands have been raised. So Alexandra, if you have you can just give the floor to the person. There's no hands
for
okay.
now, but there's a question in the chat.
Okay, can you read the question because I cannot read? I don't know.
This I think is always for Carolina. How well do different genotyping platforms agree in their results more so for the same data set? And then there is a hand up, but we'll take care of that following the answer to this question.
Thank you, Ale. I think you mean just for the same data set, having two different or more different platforms, how well these data agree. Again, it will depend on the technology and how well we can compare. It's not easy, but it can be done. That it needs a lot of bioinformatic expertise to compare, for example, an SNP chip with a DART-seq technology or a DART array versus whole genome sequencing. It can be done. We did a few years ago, but it really, required a lot of bioinformatic expertise. It's not very easy, but I think today there are a lot of applications that can be used, that we can link different technologies and anchor into the genome, and then we can compare. So there are a very nice application that I can share with you that help us to combine different applications. And mostly, yeah, agree. I mean, if we, even within DART-seq, we generate two types of molecular markers, the SNP and also the silico-DARTs, which are the present-absence variation. And I didn't explain in the presentation because you will see more detail later. So even between different markers from the same technology, we can run diversity analysis and the results are super similar. So yes, mostly comparing different technologies, the result will agree. Yeah.
Thank you, Carolina. Thank you. I think there's another question for you in the chat. Can you see it yourself? Okay.
Yes. Will it be necessary to keep everything the same while genotyping by sequencing? I don't know what you mean by keeping everything the same. I'm sorry, can you?
Ahmad, you have the right to unmute yourself. I just gave you permission, so feel free to elaborate also if you'd like to speak up.
Hello, Carolina. Can you hear me?
Hello. Yes.
Oh, okay. Thank you very much. Thank you for explaining and answering my first question. Regarding my second question, it was particularly about, for example, platform related to sequencing, the technology that we are using, the markers, the restriction enzymes that we will use. Okay, so what do you think? For example, if I have sequenced 2-3 years ago some of the samples, and today I have to sequence some more samples. So how important it is to keep everything same, and if I'm unable to keep everything same or to maintain the same standards, what do you think, how easy it could be to analyze or merge the data with previous samples? Thank you very much.
Thank you. It can be done. One special point, and I think you mentioned that, is using the same restriction enzyme. If you keep same restriction enzyme, even if your project started 10 years ago and now you are developing a new genotyping, it can be merged. And I'm happy to support and help you in the process. We do this very frequently here. So yeah, it really can be done from previous project and combining with new samples. But yes, you have to use same restriction in time. If you don't use restriction in time, it can also be done, but it's more tricky and needs more bioinformatic expertise and power.
Very well.
And I'm
Thank you very
happy
much.
to follow you on email and support you in the process.
Sure, thank you very much. Actually, currently I'm working at the Saudi Gene Bank, and I'm doing the same work, so that's why I was more interested in this.
Super, excellent.
Thank you very much.
Thank you.
Thank you, Carolina. I see a next question from Ananda. How are the data sharing policies and rules? I don't know if this, maybe you would like to pass it on. I think Claudio is still on the call, so feel free if you want to discuss that.
Well, definitely Claudio is more appropriate, the appropriate person. It's also, as I mentioned, this will be a topic in the coming day. So I don't know if Claudio wants to answer now or in the session.
Let's see if Claudio wants to say something.
Yeah, thanks a lot, Carolina. It's again an elaborated and evolving landscape in terms of what is necessary when you first of all when you when you generate the data, and when then perhaps you also make available that data through the publicly available database and repositories. We will, as was mentioned, address these in detail when we will discuss the multilateral mechanism that has been operationalized recently by the Convention on Biological Diversity, because in that particular mechanism and decision, there are certain due diligence requirements that would apply to all scientists making use of digital sequencing information, and also, of course, those of us who have data repositories who make available those data repositories on a publicly accessible basis. So I think I'll stop there, but we will discuss the details or the due diligence requirements in the session on the policy. Thank you.
Thank you, Claudio. Indeed, a sneak peek, but we won't go too much into detail because there is a whole presentation on this Friday next week. I think there's another question for Carolina in the chat. What is the approximate price for DART-seq genotyping per sample?
There is a range of prices, and it depending of course, the technology comes from the Diversity Array company based in Australia. We also provide this technology here at CIMMYT in a genotyping lab called SAGA, Genetic Analysis Service for Agriculture. In here, we did all the gene bank characterization for CIMMYT and other gene banks as well. The price can vary depending on the density of the genotyping. We have high density, mid density, low density. So depending on the purpose, will be the right, as I mentioned before, the right technology or the right density to apply. For example, if we want to discover, I will suggest middle to high density, but if you have more breeding material, more elite varieties, I will suggest low density is more than enough. So the prices may change and come from $40, $45 to $20, $25, depending on the density. So I'm happy to also support you if you have a project and if you are not sure what is the right density. It's good to discuss and understand the project so we can suggest the right options and the right cost, of course.
Thank you so much, Carolina. I think some of the following questions are all for you. Can you see them? There is one from Sevda. It's quite complex, so I'll let you read it, but I can read it out loud. Is it true that with current sequencing technologies, whole genome resequencing has become easier and more cost-effective compared to reduced representation approaches, such as GBS or DART-seq?
Yeah, I think it's more an statement, no? And yeah, you are right. But again, it depends on the purpose of the project, no? I mean, yes, whole genome resequencing is good and it's getting cheaper every day. But for example, for us, I'm managing a gene bank with 124,000 accession of wheat. So resequencing for now is not an option for us because it's still expensive. I mean, it's cheap, but it's still expensive for this big amount of samples. So we use DART-seq technology as a cheaper approach, and we can extract all the polymorphisms of the collection. So yeah, it's true. It's becoming super cheap. And if you have, for example, a small gene bank, maybe it's an option. But in our case, it's not. It was not an option. And of course, we started this project of gene bank characterization 12 years ago, because the collection, of course, is huge. So we started 12 years ago through a big project, and yeah, resequencing was not accessible or not cheap at that time. So we continue with the same technology. But yeah, definitely it's better.
I think that ties in also with the next question. So you were discussing potential use for smaller gene banks. So this question is asking, how accessible is DART-seq compared to other methods for the case of gene banks that are starting with genomics?
Well, as I mentioned, it is very accessible. And Monica, in the first presentation, show all the work we are doing with the community of practice in Latin America, for example. I mean, of course, we have providers. One of the providers is Diversity Direct Technology in Australia. And we also provide here the same technology, the same service in CIMMYT, in this lab called Saga. So yeah, again, happy to support and contact me if you have any a question or a specific project, but it is accessible. And we brought this Latin America, as Monica mentioned before, we brought this Latin American community of practice, and we generated data for different crops for national gene banks in Latin America. We are analyzing together this information. So it's not just what I like of this community of practice that we want to replicate also here is not just a service. It's not, we are not here to just provide a service. We are actually, I'm not providing a service. I'm a gene bank manager. I'm talking about a CIMMYT or that. We are here to support in the analysis and implementation of this data. Sometimes it's not, I mean, it's easy to access the technology and generate data. This is super easy. But then how we implement this information, how we apply in our routine operation, how we analyze this data. And routine operation, I mean, how we analyze redundancy of accession, how we identify unique alleles, novel alleles from our collection. So all these things is good that we discuss in this community, so we can support each other. Not only from me, but from a lot of colleagues, and even within you, I feel there are a lot of expertise that can be really powerful.
Thank you, Carolina. It seems your presentation was extremely relevant to a lot of people, which is why the questions are mostly directed to you. But I see some questions about coming back on certain topics. Since the presentations were recorded and will be shared in full, I think it's best if we avoid maybe going back on certain subjects and maybe just answer the specific questions. that are in the chat. We still have, let's say, 10 more minutes, so we can answer some more. And also, both the presenters from today have provided their emails in their presentations, so you can also reach out for more specific cases or support with certain elements of your work. So, Carolina, there's another question that I think is relevant to you. Why is it needed to resequence for a given crop or trait, as it already has been sequenced before? Also, considering costs, is this important?
Well, yeah, it's quite tricky because, I mean, taking, for example, as an example, the wheat genome. The wheat genome has been sequenced so many times, but not only sequenced many times, assemble different time, because as mentioned, the technology evolved, and before were long read, now are short read, and different type of technology that help to fix gaps, fill gaps. For example, in the old genome, we have big gaps between chromosomes. Now we can fill this gap using other technology. So not only whole genome sequencing, but also different technology that help to ensemble the genome. So I will say it's not critical to resequence if you already have sequence data. It can be analyzed in a different way, or as I mentioned, we can add different technology to improve the quality of this genome. But yeah, it is depending of the genome, no? There are some wild species or very small species with low resources sometimes. So yeah, if you already have data, it can be used, it can be improved, yes. But of course, if you have resources and you can develop new data, of course, it's also, yeah, as big project, like I'm saying wheat, because it's one of the biggest project in sequencing and resequencing, and we have now a lot of genome available. Well, not only wheat, rice, for example, maize. They are big crops with a lot of information, but some small crop with only one genome, and it's still super useful. There is
Wonderful.
another question.
Yeah.
Can you come back to DSI? We will discuss DSI a lot during the webinar, so don't worry. It will be a use component. It will be a policy component. So yeah, I think you will hear a lot about DSI during this day.
Indeed. There's also a question from a colleague from Africa Rice on a specific case. So suppose I have in silico DART data for 20 samples. Is it possible to use bioinformatics tool to perform more comprehensive SMP calls using my reference genome in order to generate a larger data set that is closer to a whole genome sequence data set?
Well, let me see if I understood. I mean, silico DART data is a very short sequence. It depends of if was previous DART, it was only 66, I think, bases, and now is up to 80 something, depending of the barcode. But they are very, very short sequence. So using bioinformatics to perform more comprehensive SNP call, yeah, of course. Using silico DART, you can extract a SNP. But if you have silico DART, you already have a SNP because it's generated simultaneously. But yeah, it can also be. I mean, this is the nice thing from this. What I like from this technology, because from using the same sequence, you can extract two types of molecular markers, no? So the SNPs and the silico DART, both are complementary because when, for example, you have a very high call rate, a lot of data in the silico DART, you have a poor SNP because you have a lot of missing data because the nature of the marker is different. So the complement between both markers it's very nice, and you can use for different purpose. I'm using, for example, for wild relative or more diversity analysis, but with different species. I'm using more silicodart because for me it's more powerful. It reveals more the difference between exotic species, for example. And for more similar, let's say land races or even improved varieties, I'm using the SNP because can look into diversity more in deep. So the combination of both, it's super powerful. I'm not sure if I answered your question right, but happy to follow up with you by email.
Thank you, Carolina. Absolutely. I think you will have a lot of work after this webinar.
Yes.
But it's great. The interaction is great. And I'm seeing a lot of questions that are coming in. There are two questions that are very similar. So in the interest of time, I would like to join them together. They're both about the communities of practice. So I think if Monica, Carolina, and even Marie-Noëlle are able to join, on this answer. So the first question is from Elizabeth, asking if is it still possible for Caribbean institutions to participate in the communities of practice that were discussed by Monica? And then there is a second question from a colleague from Mozambique asking about communities of practice and if it's open for everyone to join and to get more details. So perhaps we can answer first about the one in or more generally about communities of practice, Marie-Noëlle, and then maybe more specifically about the Latin American and African ones.
Yeah, thank you very much, Alexandra. Thank you for the great job you are doing. So I think, yes, the purpose of really setting up this webinar is frankly to have a community of practice on these important topics. So you have seen from the presentation of Monica that they already set up a very functional community of practice in Latin America. So our colleague from Caribbean is welcome to join, and they explain their approaches. So we want to transfer that model also in Africa and Asia. So this is really what we want to accomplish, so that you will be able to continue working on these tools on the molecular by learning from each other, and of course with the support of the CGIAR. So this is why we are here, is to establish the community of practice. Thank you.
Thank you, Marie-Noëlle.
So Carolina, if you want to say something.
Monica is here. Do you want to answer this question, Monica?
Monica, sorry.
Yes, but sorry, can you repeat the question?
Yes.
The thing is, I'm driving.
Oh,
I can answer. Don't worry, I can answer. It's just, yeah, don't worry. The community of practice is open, of course. We developed this community of practice for Latin America. Currently, we have 15 countries represented, mainly national gene bank from all these countries. It's totally open. Actually, we are happy to receive new members of the community. As Monica explained, we started in 2021 and we continue until now, including new, we started with genomic information, but then we continue with different topics depending of the interest of the community. So yes, definitely. The only thing is, in this case, in the Latin American community, we received the request to be a community in Spanish, because the people feel more comfortable with the language and are more open to discussions and learn more in deep. So yeah, this is the only thing that we request to be fully in Spanish. But again, we are trying to develop this community in different regions with their language. And so yeah, definitely it's an option.
Thank you, Carolina. I think since we're almost running out of time, I would take maybe a couple more questions, and then I would suggest the participants to reach out individually if they have specific cases, or perhaps try to catch your facilitators in the next presentations that they will be given. So there is a question again on genome sequencing, is for Carolina, I assume. Is genome sequencing and characterization available for external projects? So for example, cooperation to development or even for privates?
Yeah, of course. I mean, we don't. I'm not sure if I understand. It's if we provide this service, or
I think that's the question. Yeah,
right. We focus in DARSIC technology here at CIMMYT. There are others, of course, service provider, and we can also support to guide you and give you options. But for now, we don't do whole genome sequence here. Even if we have the technology, we try to focus in our expertise is more in germ plants characterization and breeding program, and that's why we use more genotyping technology than whole genome sequencing. But yeah, there are plenty of service provider that can help you with whole genome sequencing.
Thank you, Carolina. I think one last question or more consideration on this global topic is coming from Onis Musa, our colleague at the Zimbabwe National Gene Bank. So he's making a point, which I think it would be great for discussion as well. So as a research institution, gene bank in a small developing country, we're just trying to adopt these molecular technology approaches, comes with a cost in terms of establishing facilities and maintenance, also considering how fast advancing they are. Would it be wise to establish those facilities as opposed to outsourcing them from already established institutions?
And this is a great question. I will say you raise a question that many gene bank or many institutions are having now. I will say don't waste $1 in a lab. And I'm talking with experience. We did, and that's why we are a service provider now. But I think it's not a good approach because they are very expensive technology to set up. We are talking about $1 million sequencer for a small institution or for a small project. Doesn't make sense. Invest these resources in outsourcing the sequencing, and invest this in bioinformatic equipment and expertise. I think this is where many centers and many, especially small centers, need to invest more in expertise and bioinformatics, because today it's very easy to access this technology in service provider. So I mean, depending of why we developed this technology here at CIMMYT, because it was a big project. We have two of the largest gene bank in the world. So at that time, makes sense to develop and create this platform here. And now we open, well, not now, like 10 years ago, we open as a service as well for other crops. But I will say it's not a good decision to invest in a new lab because the technology are very expensive. I mean, you can invest in library preparation, for example. No, you need some PCR, small equipment, but not a sequencer that in 2-3 years it's old and you have a new one. Just to give you an example, we had previously a high-seq and now we have a NovaSeq. A high-seq, it was great. It worked for 5-6 years, but then Illumina company say, I'm not supporting any more high-seq. So it's sitting there doing nothing, and it's still working very good, but we don't have support. We don't have any more reagents. So yeah, you have to, if you are a center that run thousands and thousands of samples every year, maybe it's good, yes, but you need to really think in the investment is supported by the number of samples by year. If not, invest all these resources in outsourcing and invest in expertise in bioinformatics in your center.
Okay, thank you, Carolina, for the nice answer. I can see that we have only one question on community of practice again. So the community of practice will be open to all the participants. Please be engaged, respond to the question, listen to the recording, try to understand all the key concepts coming from each of the presentation, so that we will use those knowledge now to build the community of practices in each of the regions. So I think with the response of Carolina, we are coming now to the end of this webinar series today. And so I would like really to thank all the presenters who really join me, please, so that we can upload all the presenters for today. And thanks, Carolina, thanks to Monica for the nice presentation. Thank you all participants for your presence, and also for the nice, excellent question that you were able to raise. So we will see tomorrow at, please let us, we see tomorrow at 1 p.m. European time, 1 p.m. European time. Please let us start on time so that we have enough time also to respond to the question you will be asking. So thank you so much for being there, for coming so many to attend this webinar, and see you all tomorrow at 1 p.m. European time. Thank you. Bye-bye.
Thank you. Bye-bye.